pop quiz. Which process allows your body to grow? Mitosis or meiosis? Lock in your choice. It's mitosis, the cell cycle that makes genetically identical cells for growth and repair. In this video, you'll learn the stages of the mitotic cell cycle, the checkpoints that control them, and how mitosis compares with meiosis. First, a quick map. A cell's life follows a repeating cycle of growth, DNA replication, and division, called the cell cycle. In the mitotic cycle, the division produces two genetically identical daughter cells. During interphase, the cell prepares for division. In G1, the first gap phase, it grows and checks for DNA damage, and at the G1 to S checkpoint, it pauses if any errors are detected. Next, in S or synthesis phase, the cell copies its DNA to make sister chromatids. Then, in G2, it makes the proteins needed for division and pauses at the G2 to M checkpoint to verify DNA integrity. Now comes M phase, or mitosis. It begins with prophase, when chromosomes condense, the nucleolus fades, and a mitotic spindle made of microtubules forms. These microtubules are protein fibers that attach to kinetochores on each chromosome's centromere. Next is metaphase, when all chromosomes align at the cell's center to form a metaphase plate. Each chromosome is pulled from both sides to ensure balanced tension. Before moving on, the cell pauses at the spindle assembly checkpoint, or SAC, to confirm that every chromosome is correctly attached to the spindle. Curious how much you're remembering? Your personalized practice test, flashcard set, and study guide are all waiting for you. Just tap the link now and keep watching. Future you will thank you. Once that checkpoint is satisfied, the cell enters anaphase. Here, the centromeres split, and the sister chromatids are pulled apart to opposite poles. This is followed by telophase, when nuclear membranes reform, nucleoli reappear, and chromosomes uncoil back into chromatin. The cell now looks like two nuclei sharing one cytoplasm. Finally, cytokinesis splits the cytoplasm. In animal cells, a contractile ring of actin forms a cleavage furrow. Meanwhile, in plants, vesicles meet in the center to build a cell plate that develops into the new cell wall. The result? Two genetically identical daughter cells with the same chromosome number as the parent cell. That's why mitosis is called an equational division. By contrast, meiosis is a reductional division that produces gametes with half the chromosome number. Mitosis creates genetic copies, whereas meiosis generates variation. For example, organisms such as amoeba and hydra reproduce asexually through mitosis, producing offspring identical to the parent. In humans, mitosis replaces worn-out cells and heals tissues. Once division is complete, each daughter cell enters a new G1 phase and the cycle begins again, ready for the next round of growth and repair. Taken together, the G1 to S, G2 to M, and spindle assembly checkpoints maintain order and accuracy through each division, preventing errors that could lead to disease. To recap, mitosis produces two identical cells through prophase, metaphase, anaphase, telophase, and cytokinesis. Checkpoints at G1 to S, G2 to M, and metaphase ensure safe, error-free division. Ready to quiz yourself with the content covered in this video? Check out the link in the description where you'll find Quizlet content made just for you.